We are continuing our discussion of how cells divide. And we have talked about how the body cells divide and why and when the body cells divide. Remember, division of the body cells is called mitosis. And it's important for us to make the distinction between mitosis and what we are now going to talk about, which is called meiosis. So mitosis was division of the somatic cells. All the cells of the body except for eggs and sperm. And the goal of mitosis was to produce two genetically identical cells. Why do you think we need those cells to be genetically identical? Why would it be a good idea for all the cells of our body except our eggs and sperm to make proteins the same? Well, because if we didn't make proteins the same way, which remember that's what DNA codes for. DNA is coding for how to make a protein. Put this amino acid first, this one second, this one third. We want that consistency. We can't make each protein differently with a different amino acid sequence. We would just be this big jumbled up mess that didn't work. You wouldn't have a living organism. You need all the cells of the body to have the exact same recipe for how to make a protein. We need the same DNA. We need the same control systems in all of our cells. So the goal is to take a diploid cell, go through one division, and produce two genetically identical diploid cells. Remember, diploid is 2N, which for humans is 46 chromosomes. The goal of meiosis is going to be different. When we make gametes, first of all, we don't want them to be genetically identical. That is a very, very important distinction. Why do you think we don't want all of our offspring to be genetically identical? We need genetic variation in a population for that population to be sustainable over a long period of time. We know that when we reduce population sizes of organisms down to a very small number, and we don't have that genetic variation, there sometimes aren't individuals around who can survive certain changes in the environment. It's important to have that genetic variation for the long-term sustainability of a population. Genetic variation in your offspring is very important. So that's going to be one goal of meiosis. When we're making our gametes, we want them to be genetically different. So meiosis is going to be division of the gametes, the eggs and sperm. And we don't want them to be genetically identical. The other thing is, do we want them to be diploid? Well, that's a good question. What would happen if we had a 2N cell that was an egg and a 2N cell that was a sperm? How many would we end up with in the resulting offspring? 4N. And then this one has babies with somebody that's 4N, and it ends up being 8N. And before you know it, we have millions of chromosomes at this point in human history. We know that's not what happens. So the other goal of meiosis is to reduce the chromosome number. So when we're making these gametes, we're going to take a cell that's 2N. Why are we starting with a cell that's 2N? Because you were made from an egg that was haploid and a sperm that was haploid, which made a diploid cell. And then every cell of your whole body, including the precursors to your sperm or your eggs, was a diploid cell. And now we have to take that diploid cell, just as we used it to make a stomach cell, a skin cell, a brain cell, now we need to make gametes. We need to make sperm cells or egg cells. So we're going to go through 2 rounds of division. In the first round, we're going to already get down to the haploid number, and then each of those is going to go through a division. So at the end of meiosis, we started with a diploid cell. We're going to end up with 4 haploid cells, and they are all genetically different. So the story I'm going to tell you about meiosis today is going to explain how we get down to 4 haploid cells from this original diploid cell, and also how we produce this genetic variation in our gametes. I'm going to write this statement on the board. Genetic variation in gametes. This is a very important concept in biology. You want genetic variation in your gametes. So let's just look real quickly at the screen, and I'm going to remind you of some terms that we already discussed in the mitosis lecture. I'm not going to go into as much detail, so if these terms don't make sense to you, please rewatch that lecture so you get the relevant terms. This is a karyotype. It's showing a full array of all 46 human chromosomes, so 23 pairs. Remember, pairs 1 through 22 are called the autosomes. These are not the sex chromosomes. Chromosomes 1 through 22 are called the autosomes. And then the 23rd pair, those are called the sex chromosomes. A very important distinction I need to make here. For some reason, these terms are sometimes confusing to students. Sex chromosomes and gametes are not the same thing. Okay, an egg or a sperm, let's pretend you're female and you have an egg with 23 chromosomes. That has all 23. That is an egg. That is a gamete. When we talk about the sex chromosomes, we're just talking about that 23rd pair. You're either XX or you're XY or you're some variation on that theme. But sex chromosomes is only talking about these two. Okay, a gamete, an egg or a sperm, has one of each of all 23. It's very different. These terms are different. Okay, another term that's very important to remember, homologous chromosomes. Homologous chromosomes are going to be a very important part of this story. Remember, homologous means speaking the same language. These chromosomes not only have the same number, but they code for the same traits in the same location. So these two number 1s are homologous chromosomes. These two number 12s are homologous chromosomes. In this picture, each chromosome has 2 chromatids. Remember, we start off with 1 chromatid for each chromosome. And in the S phase of the cell cycle, we made an identical copy and attached it with some cohesin proteins. When they're attached, they are sister chromatids, and they are genetically identical. When they're not attached, let's say this is mom's chromosome number 1, and this is dad's chromosome number 1. These are sister chromatids, they're attached. These are sister chromatids, they're attached. This one of mom's and this one of dad's, these are called non-sister chromatids. They code for the same traits in the same location because they're homologous chromosomes, but they are not genetically identical. No way for mom and dad to be genetically identical when we're talking about humans. So these are non-sister chromatids. That's important because we're going to do some gene swapping in prophase 1 and meiosis between non-sister chromatids. Sister chromatids are genetically identical. No point in doing gene swapping there. But one of the ways we're going to get genetic variation in our gametes is we're going to do some gene swapping. So these homologous chromosomes are going to pair up. So those terms are very important to remember. So I just want to quickly take you through this overview. Just so you remember these terms. Again, if this is a homologous pair, one from mom and one from dad, so in this picture they're showing mom's as being red and dad's as being blue. These would be sister chromatids because they're attached. This one from dad and this one from mom would be non-sister chromatids. Here's what we're going to do in meiosis in a nutshell. This is without doing gene swapping. This is just the basic story. We started off with a cell that had a full array of chromosomes, that each one of those chromosomes just had one chromatid. Okay, and then what we did is in the S phase of the cell cycle, we replicated that DNA, and now they all have a sister chromatid attached. So each one of these would have a sister chromatid attached. There's a homologous pair, one from mom and one from dad. What we're going to do is in the first round of meiosis, we're going to get these two together. They're going to do some gene swapping so that now they're not just purely mom's DNA and purely dad's. They're going to be a mixture of the two. And in the first round, we're going to take this pair and separate them into two separate cells. Imagine that these are pretend animals going onto the pretend Noah's Ark. Okay, so a pair of elephants. In the first round, one elephant's going to go into one boat and one elephant's going to go into the other boat, completely intact. Both chromatids stay together. And then in the second round, we're going to separate the sister chromatids. That's how we're going to end up with 4 cells that are haploid. Imagine for each of our 23 pairs of chromosomes, here's our homologous pair. We have 4 chromatids, 1, 2, 3, 4. We're going to separate all 4 of those into 4 separate cells. And they're not going to be their original DNA, they're going to be a mixture of mom and dad's DNA all together. So that's the basic story of meiosis. 2 goals: reduce the chromosome number down to haploid and create this genetic variation in our gametes. So now let's go through the details. Let's go through the steps of what happens at each stage of meiosis. Now let's start looking at the details of what happens at each stage of meiosis. We're going to go through meiosis 1 and then meiosis 2. And here it is, all in a nutshell, all on one slide. Here it is. So we're going to start with a cell that has 46 chromosomes, or the 2n number. And the first thing that's going to happen that's very different from mitosis is all of the homologous chromosomes are going to pair up. In mitosis, they don't need to pair up. They can just be random all over the cell. It doesn't matter because all we're doing is connecting each sister chromatid to a microtubule so we can pull them into 2 separate halves of the cell. The 2 number 1s might end up next to each other or they might not. It doesn't matter. But in this case, we want the 2 number 1s to get together and the 2 number 2s and so on. We need them to all pair up. Again, like the imaginary animals going onto the ark. Everybody needs to pair up with his buddy. So the one from mom and the one from dad are going to come together. And that's the first event that happens in meiosis 1, prophase 1. You should already kind of know how these events go just based on what happened in mitosis. Looking at what happens in prophase, metaphase, anaphase, and telophase, it's very similar in terms of how things line up and connect, but there are some subtle differences that are important to note. So here we are, meiosis 1, prophase 1. Homologous chromosomes pair up. This did not happen in mitosis. They pair up and they're going to do something very significant, and that is they're going to do some gene swapping. They're going to do what's called synapsis, crossing over. There are a lot of terms associated with this process. Crossing over occurs, and this is really gene exchange between mom and dad's DNA. So at the end of prophase 1, rather than having one chromosome be just mom's DNA and one be just dad's DNA, we now have chromosomes that are a blending of both. This is the first way that we get genetic variation in the gametes. So this is what's going to give us genetic variation in the gametes, in our eggs and sperm. So prophase 1 is when this occurs. When the 4 of those are together, and when I say the 4 of them, I mean the 4 chromatids. So let's assume this is mom's chromosome number 1. And then dad's chromosome number 1. Yeah, that's not writing. So here's dad's chromosome number 1. And when those two get together, it's essentially 4 chromatids together when they pair up. That's called a tetrad. That tetrad is going to do some gene swapping. So when we're done with this whole process, this is going to be a mixture of mom and dad's DNA, and this is going to be a mixture of dad's and mom's DNA. And it's all random how it occurs. So let's have this mirror because they swapped the exact same genes. So right now, if I separated these 4 chromatids into 4 separate cells, you can see how each of those cells would be getting a different version of that DNA. That is the major way we're going to get genetic variation in our gametes. Crossing over this gene swapping that occurs in prophase 1 of meiosis 1. A very, very significant event. The events, other than that, are pretty similar to what we had in mitosis. The only difference is that here in prophase 1, what's going to happen is because we have these homologous pairs that have teamed up, I'm just going to draw 2 pairs of chromosomes here. So these are going to be the tetrads. So moms and dads have paired up, they've done their gene swapping. I'm not going to show them stripy at this point. Now what happens is the way the microtubules attach has to do with the goal of meiosis 1. The goal of meiosis 1 isn't to pull sister chromatids apart. The goal of meiosis 1 is to separate that homologous pair. So take this whole chromosome from mom with both of its chromatids, and this whole one from dad with both of its chromatids, and move them into 2 separate cells. So that's how the microtubules are going to attach. They're going to attach so that each member of the homologous pair is attached to microtubule from an opposite pole. Okay, so each homologous chromosome, sorry, I'm over in there, this chromosome, is attached to a microtubule. And they're attached to opposite poles. Think about the goal. The goal is to pull them into 2 separate cells. So we need to attach them that way. Okay, when they're aligned at the equator like this, we're at metaphase 1. Okay, so they attach to microtubules here in prophase 1. By the time we get to metaphase 1, they're aligned at the equator. Okay, so here they are attaching, everything pulls. Now they're here at the equator. Anaphase 1, sister chromatids don't pull apart in anaphase. Remember, we're just trying to separate the homologous pair. So anaphase 1, homologous chromosomes separate. So that would look something like this. Using the same color scheme we had before. We're going to have a whole intact mom's chromosome and the whole intact dad's chromosomes moving to 2 separate cells. But guess what? Here's another twist to the story. When these aligned on the metaphase plate, it might not have flip-flopped this way. When that tetrad lines up on the metaphase plate, we might not have all of dads facing one direction and moms facing the other. It might have been like this. It might have been that moms ends up here and dads ends up here. And imagine that for 23 pairs of chromosomes. This is the other way we're going to get genetic variation. Because it could have ended up like this when we divide it in telophase, or it could have ended up like this. And add to that the fact that none of these really are purely mom's or purely dad's DNA anymore. Remember, they're going to all be striped. They're going to all be a mixture of random gene swapping that occurred. So imagine that when they line up on the metaphase plate, each pair can flip-flop either direction. And imagine that for 23 pairs of chromosomes. It's all random. You might end up with a cell that's 80% dads and 20% moms, 50-50, 90-10. It's all random each time. That is called independent assortment. How the chromosomes, how that homologous pair, that tetrad, lines up on the metaphase plate and then assorts independent of the other pair. Independent assortment. This is going to give us genetic variation as well. So crossing over in prophase 1 is going to give us genetic variation, and this random alignment of the chromosomes on the metaphase plate of metaphase 1 is going to give us genetic variation as well. Very, very important to understand. So then we pull them apart in anaphase. Okay, and then in telophase 1, we end up with 2 cells, each of which just has the n number of chromosomes. So here we still had 2n. Now each of these just has 23 chromosomes, one of each pair, but each one has 2 chromatids. So looking at how I've drawn it in this last example, we'll just go with that for telophase. So one of mom's and one of dad's. Now what we need to do in meiosis 2 is pull those sister chromatids apart. So that's how they're going to line up now. So looking at prophase 2, now the microtubules are going to connect to the sister chromatids, just like they did in mitosis. When everything's aligned on the metaphase plate, that's metaphase 2. Sister chromatids pull apart in anaphase 2, and then we divide those 2 cells in telophase 2. So let's quickly go through those steps. So each of these cells is now going to enter, come on, you can do it, prophase 2. In prophase 2, what's going to happen is each of these cells is going to have a situation where microtubules attach to each sister chromatid. So I'm just going to do this top one. Same thing will apply to the bottom one. Okay, so each sister chromatid attached to microtubules from opposite poles. When they're aligned at the equator, then we're at Metaphase 2, I don't need to draw that on the board, right? You can get that image. Anaphase 2. Sister chromatids separate, just like in mitosis. The only difference is that we started off with a haploid number of chromosomes. So here's our cell. The sister chromatids are going to pull apart. Okay, and then as we move into telophase 2, for each one of those cells, we end up with 2 new cells. Each one has 1 chromatid from the original chromosomes. Okay, and we do that twice. We do it for this one too. So this one's going to go through that same process. So if that one goes through the same process, we're going to end up with 2 more cells, each of which has 1 chromatid from the original. 4 haploid cells. And remember, all of these chromatids are actually a mixture of mom and dad's DNA. And they're all going to be different from each other. Imagine that for 23 chromosomes. All 4 of these are all genetically different from each other, and they're genetically different from that cell we started out with. All genetically different. This is why if you have a sibling, same parents, you might look kind of like each other. You're going to have some similar characteristics. People will most likely, in many cases, be able to tell you're related to each other, but you don't look exactly the same. Unless you're an identical twin, then you look more the same. Even those don't look exactly identical, but you look similar. But the reason all of your siblings look a little bit different from you is because of this. So remember, 2 times that we created genetic variation. One was during prophase 1, this crossing over, this gene swapping. And then the other was just this random alignment. You can see in this example, 2 of the red ones are facing up and 1 is facing down. That could have been completely different. So those are the major steps of meiosis. Again, I didn't give you as much detail as in mitosis because the steps are somewhat similar. In prophase, we're always attaching microtubules, and they're going to pull until we get aligned at metaphase. Things separate at anaphase, and telophase is going to give us 2 new cells. The only difference is that now, remember, in prophase 1, we're going to align everything and attach microtubules such that a microtubule is attached to each intact chromosome of the homologous pair. The goal is pull the homologous pair apart. And also this important crossing over takes place. So anaphase 1 is just separating the homologous chromosomes. The homologous pair separates. Anaphase 2, sister chromatids separate. So in thinking how things attach and connect during prophase, think about what the goal is. We're either separating the homologous pair or we're pulling sister chromatids apart. So just to compare again, mitosis and meiosis, and then we're going to look at crossing over again one more time just because it's such an important concept. So in mitosis, everybody just lines up independent of each other. 1s don't get together, 2s don't get together. They might just happen to end up next to each other randomly, but it's just random alignment. This, in meiosis, is not random alignment. Everybody's paired up with their homolog. So round 1, homologs separate. Round 2, sister chromatids separate. And you can see that these 2 cells are genetically identical to each other. These 4 are all genetically different. Also here we ended up with 2 diploid cells. Here we end up with 4 haploid cells. If you imagine that each of these chromatids are going to end up in 4 different cells, that's what happens. The details here we've already talked about. There's no crossing over in mitosis. You only have 1 random division. You only end up with 2 daughter cells, not 4. Mitosis, remember, is for development, growth, replacement of damaged cells, replacement of cells that are past their lifespan, repair of damage. That's what mitosis is for. Meiosis is just for making eggs and sperm. Those are the only cells that go through meiosis. This just shows you the different possibilities. We talked about how things align at metaphase. I don't have metaphase drawn on the board anymore, so I'll just show you here. So it's just showing 2 different possibilities. Here, all of one parents are facing one direction and all of the other parents are facing the other. They've turned this sideways instead of North Pole, South Pole. It's kind of east-west. So here's the other possibility, that it was 50-50. So when we split this the first time, we ended up with 1 cell having all of one parent's DNA and 1 cell having all of the other parent's DNA. That's going to be a very different result at the end. So this doesn't have much genetic variation. This already has genetic variation without even crossing over. So remember, that's called independent assortment. It happens during metaphase of meiosis. So crossing over is what creates this genetic variation. And here we go. Here are the chromatids. It's very important to note, so let's again pretend this is chromosome number 1. Here's mom's DNA. I'm not going to take the time to color this in. Here's dad's DNA. When crossing over occurs, does it occur between sister chromatids or non-sister chromatids? What would happen if sister chromatids did gene swapping? Remember, sister chromatids are genetically identical to each other. At the S phase, we made an exact copy. What would be the point of sister chromatids doing gene swapping? There's no point to that. You're swapping the exact same gene for the exact same gene. So it's non-sister chromatids that do this gene swapping. Non-sister chromatids. They're the same homologous pair. They're both chromosome number 1, or they're both chromosome number 2, or they're both chromosome number 3. But it's the non-sister chromatids that swap genes in prophase 1. No point in having sister chromatids swap genes. So that's what we're looking at here. Non-sister chromatids are going to do some gene swapping. This is an awesome picture. This shows you that's exactly how it happens. It really does. We didn't just make that up in this cartoon picture. Now, all 4 of those chromatids look different from each other and different from how they started out. Then in anaphase 2, when we start to pull them apart, you can already see that our 4 cells are going to look different from each other. So 2 ways we get genetic variation. I know I keep repeating this, but it's such an important concept. We do not want all of our gametes to be genetically identical. We need genetic variation in a population for that population to survive long term. Crossing over in prophase 1, and then this random alignment called independent assortment during metaphase. And that's just a close-up again of that crossing over occurring. So that's how we make eggs and sperm. We're going to talk about what can go wrong when we talk about inheritance of traits. We'll talk about how things can go wrong during meiosis and what the results are of that.